Okay, good morning. I hope fall break went well. So thanks for being here after that. This morning I'm going to be mainly talking, probably entirely talking, about the DNA viruses and finishing off on the DNA viruses, we hope. At the end of last lecture, we had started to talk about herpes virus. Remember, much bigger virus physically, but probably sort of more important when you come to look at what they do, a much bigger genome. And we talked about how herpes virus fuses with the plasma membrane and how, because it can fuse at physiological pH, it fuses with the plasma membrane on the way into the cell, but later on those fusion proteins can also facilitate fusion between cells. And so these are cells that cause cells to fuse and you get syncytia. And you see that for the three families that fuse with the plasma membrane. The paramyxoviruses, the herpes viruses, and HIV typically show these syncytial formation. Okay, so this is just an overview of the life cycle so that you can put it all together at the end. But I'm going to talk about each step individually, so I'm going to start at the top of here and work my way down the diagram. So here we have the herpes virus binding to the cell surface and fusing with the plasma membrane and letting its nuclear capsid enter the cytoplasm. Okay, so that's shown down here. And one thing is, yet again, the nuclear capsid takes advantage of the host cell, hitches a ride on the microtubule system, is delivered, we assume, to a nuclear pore because you can find empty nuclear capsids at nuclear pore, but you don't find them inside the nucleus. So it appears that the DNA exits the nuclear capsid and enters the nucleus as DNA, maybe with a few associated proteins. One thing here is to notice also, we said, that herpes virus is a much more elaborate virus than the ones we've talked about so far. And I mentioned there was a layer of protein, it's got some structure to it, in fact, between the nuclear capsid and the membrane. And we call that layer the tegument. And it has some proteins in it that are very important in the herpes virus life cycle. I'm not going to bore you with all of them, but I'm just going to bore you with one of them. This one here, VP16, VP means it's a virion protein, i.e. it's a structural protein. It was present in the virus particle. And that protein is released, the tegument proteins are released into the cytoplasm, and they go off and do, some of them go off and do certain jobs. This VP16 has got a nuclear localization signal on it, so it goes into the nucleus. So now you've got the DNA into the nucleus with some associated proteins, and you've got this VP16 in the nucleus. And VP16 turns out to associate with host cell transcription factors. The host cell transcription factors could recognize some of the early genes of herpes, but with very, very low efficiency. And remember, at this stage, you've got one DNA molecule in the nucleus. And you really want to get pretty efficient messages if you're going to make any decent amount of protein. One DNA is not, you want it to be copied into multiple messages and reasonably fast. So this VP16 protein associates with some host cell transcription factors and makes them work very well on the early genes of herpes virus, so that you get a lot of transcription at this early stage. And this increases the transcription from the SOSA alpha promoters. Herpes virologists appear to be very heavily influenced by their Greek alphabet. And you'll come, I suspect, when Dr. Gangemi talks to you about the herpes virus families, he's going to talk to you about the alpha herpes group and the beta herpes group and the gamma herpes group. When it comes to the genes, we have the alpha genes and the beta genes and the gamma genes. Alpha genes are the immediate early genes. In other words, those that are made immediately upon the virus entering. So they're what you called immediate early when we talked about adenovirus. Beta genes are the so-called delayed early. You need the immediate early genes in order to switch on the beta genes. So those were what we called delayed early. They are, so both the alpha and the beta are the early genes. It's just that the alpha are earlier than the beta. And then DNA synthesis will start, and you can start making the late genes. And the herpes virus people call late genes gamma genes. but it's just, unfortunately, a different nomenclature. I think there's a gradual effort to move these into the same nomenclature as the other viruses, but it's meeting a lot of resistance, as you'll see when you look at the textbooks. Okay, so VP16 goes to the nucleus. It interacts with host cell transcription factors so that they see the immediate early promoters. And then they make up messenger RNAs, which are cap methylated, polyadenylated, because they're using the host cell RNA polymerase again. If you go to the nucleus, as I pointed out before, the RNA polymerase is there in all cells that have a nucleus. They all make messenger RNA. So if these viruses get to the nucleus, there's no particular great advantage that one can see in them not using the RNA polymerase. And as I pointed out, since it works as a complex with the capping and methylation enzymes and with the polyadenylation enzymes, it seems difficult to use any of those aspects of RNA synthesis individually because it's all tied together. So what seems to happen is the RNA polymerase functions as a factory, caps, methylates, and polyadenylates the RNA as it makes it. And if you just have an RNA that's floating about in the nucleus and hasn't been fed into the machinery at the right entrance point, it doesn't get all of those modifications. So if you want your RNA to be modified, you have to have the host RNA polymerase to make it. So if you go to the nucleus and you don't use the RNA polymerase, you have to provide all those functions as well. And it seems, as far as we're aware, none of these RNA viruses have chosen that route. It would take a lot of coding information for something that's present in all cells. When it comes to DNA synthesis, that's not present in all cells. So there's an advantage of taking your own DNA synthesis machinery in because you're not having to rely on causing this cell to divide and make its DNA synthesis machinery, or at least go into S phase. So you make these immediate early proteins, and I'm not going to go into what all of them do, but one of their important functions is to enable the beta protein promoters to be recognized. So they include proteins that modify transcription and enable those beta proteins to be recognized.